Hello everyone, I am Kintal Kumar studying B Pharmacy third year in Maharaja College of Pharmacy. Today I am giving a seminar on the topic Southern Blotting Technique. It is a molecular biology technique used to detect a specific genetic sequence in a genomic DNA. This technique was introduced by Sir Edwin Sutherland in the year 1975. Now I will go with in detail process. See here, Southern blot. Genomic DNA is first isolated and treated with restriction endonuclease. Now, the fragments generated. We all know what will happen if we treat a DNA with restriction endonuclease. The fragments generated by restriction digestion are separated by agarose-gel electrophoresis. There many fragments were produced due to restriction digestion. A smear is observed on the agarose gel plate. Agarose gel is treated with alkali to separate the double stranded DNA in single stranded DNA. See here, alkali which is NaOH. Then a single stranded DNA is then transferred on the membrane by placing the membrane on the top of the gel. Here the membrane is nylon membrane. The transfer usually occurs by capillary rise action. Here, once the transfer is completed, membrane is heated at 80 degrees for 2 hours or exposed to UV light. Here, ensured the DNA is permanently attached with the membrane. The next step is of detection of specific desired fragments by using probes. We are taking a hybridized solution which contains probes. The probes has nucleotide sequence complementary to the desired DNA which is to be detected. After hybridization, the excess of probe is washed out and an X-ray film is placed upon the membrane to perform autoradiography. Autoradiography, it shows the presence or absence of desired DNA fragment. Now we'll go to applications. Presence or absence of gene. We all know that when an restriction endonuclease enzyme is added to a DNA sample, the restriction endonuclease will cut the DNA at specific sequence. It cleaves the upstream and the downstream of the DNA sample. And presence of band, if after the experiment if a band is present on the autoradiograph, that represents the presence of gene C, which was marked with yellow circle, and if there is no mark, then it represents the absence of gene. This is the technique. We also use it to determine the copy number of genes. See, difference in size of gene is easily spotted in autoradiograph. You can check, which was circled here. These two genes have similar size. Now we'll go with other applications. See here, the other applications include RFLP, restriction fragment length polymorphism, and the AFLP, amplified fragment length polymorphism. Later we will discuss the applications in detail. Now we will go with simple view of Southern blotting technique. Here you can see, here it's given RNA or DNA. In case of Southern blotting technique, we can determine the genomic DNA. In case of Northern blotting technique, we can determine the RNA. See, first the DNA sample is taken and by using restriction endonuclease, it was cut into pieces and then placed on the agarose gel plate, and then by the addition of alkali, we separate the DNA fragments by their size, and a nylon membrane is placed upon the agarose gel plate, and that apparatus is both the plates are placed in a beaker with salt solution, and Wattman filter paper is placed upon the nylon membrane and a weight is placed Below the agarose gel plate, a sponge is placed. Through the sponge, the solution is passed through it by capillary action, and it will take the solution takes the DNA fragments from gel plate to nylon membrane. Later, it was removed. The nylon plate was dried for 2 hours or exposed to UV light for few seconds, and then the nylon sheet is placed in hybridized solution which contain probes. These probes have nucleotide sequence complementary to the desired DNA which is to be detected. After hybridization, the excess probe is washed out. An X-ray film is placed on the membrane to perform autoradiography. This is the simple view of Southern blotting technique. Now we'll go with applications in detail. Southern blotting technique is used to identifying specific DNA in a DNA sample, and it is also used in the preparation of RFLP maps, restriction fragment length polymorphism maps, and also in case of detection of mutations, deletion or gene rearrangement in DNA, and also for criminal identification and DNA fingerprinting, and in case of detection and identification of trans genes in transgenic individuals, and in also mapping of restriction sites, and also in case of for diagnosis of infectious diseases. These are the applications which we are using the Southern blotting technique, and we include this progenesis of cancer and prenatal diagnosis of genetic diseases, and the last but not the least, determination of the molecular weight of a restriction fragments and to measure relative amount in different samples. These are the applications which we are using the Southern blotting technique. And here the references which I took this topic from these websites. I took this photos and the video. Thank you guys. Thank you for this giving wonderful opportunity. Thank you.